Hello students, in this video we will discuss about vectors. What are vectors? Vectors are the vehicles which takes our foreign DNA into the host cell. For example, you have to go somewhere from one place to another. So you need a vehicle to go your destination place. So you must take a vehicle or transportation to go that place. In the same way, the foreign DNA or the alien DNA needs a vehicle to go into the host organism. So this vehicle is known as the vector. So vector we can define that it is a molecule, it is a transporting vehicle of the DNA molecules. And there are two types of vector. First is expression vector and second is cloning vector. Expression vector is that which is used to express the genes so that our required or desired protein has been formed. And in cloning vector, that is, as its name suggests, cloning, that is it is used to make the copies of that desired or that foreign DNA. Cloning means clone of making a copy of the same DNA. So cloning vector is used to make the copies of the DNA and expression vector is used to make the protein. So but expression vector is not in our course, so we will focus on cloning vectors and what are the features of the cloning vectors. So a cloning vector must able to replicate in the host organism. Obviously, the cloning vector, its main function is to make clone of its own DNA, makes copy of the DNA so that we can get our desired DNA in multiple number. And it must have restriction sites. Why it must have restriction enzyme sites? Because these sites will allow to cut the vector DNA. This is a vector DNA and it will cut from these sites so that we can incorporate our foreign DNA or alien DNA. And it must have genetic marker. Genetic marker or the selectable marker are those which help us to screen the transformant cell. Now what is transformation I will tell you. And it must have low molecular weight and high copy number. That it means it replicates and will make a high copy number. It must have restriction sites to cut down the DNA, vector DNA, and the sites to ligate our foreign or alien DNA. This is the PBR322. This is the first genetically modified vector, and these vector contains selectable markers as well as these restriction sites, okay, and the ORI, that is origin of replication, from where the replication process starts. This is a PBR, the first the first vector, and its name has been derived like P means plasmid. What is plasmid? Plasmid are the extra chromosomal DNA which is present in prokaryotes, and this extra chromosomal DNA is circular in shape, and some for the construction of these PBR, this vector, that is PBR322, two plasmid has been modified and a desired vector is constructed. Why it named is desired vector? Because it contains a selectable marker as well as ORI and restriction sites. So a good vector or ideal vector is that which must have these three properties. So the PBR322 has been constructed by using two different plasmids, and hence this has been made. And so is the nomenclature. P means the plasmid, BR is the name of the scientist, and 322 is the strain number of this vector. So the plasmid, now we use the plasmid like for the vector we use plasmid. The plasmid can be of bacteria, animals, bacteriophage and plants. So the plants plasmid are used in some cases in which the vectors for cloning genes in plants. So the famous vector which is used for cloning plants, it is TI plasmid vector. So what is TI? It is these plasmid has been isolated from Agrobacterium tumnificans, and this bacterium caused gall formation in the tumor, or the tumor formation in the roots of the plant. And by modifying this plasmid, this TI plasmid, vector DNA is made, which takes a foreign DNA into the plant host. Okay, so here you can see this is the origin of replication, from where our foreign DNA will be ligated and replication process will start. And this is tDNA region, that is transfer DNA region, means this region will be transformed into the host cell or into the plant cell. The tumor inducing, that is TI plasmid, which is present in Agrobacterium tumnificans, has been modified into this, that is cloning vector, which is used to deliver our genes of interest in a variety of plants. Apart from that, retroviruses has also been disarmed, or has been modified so that it deliver our which deliver our required genes into animal cells. These are some features of the cloning vector. A cloning vector must have origin of replication, that is ORI, the site from where the replication process starts, a selectable marker which is used to screen the transformed and non-transformed cells, and the cloning site, so that we can get a multiple copies of the required DNA of the foreign DNA. This is a diagram, or this is a figure of the PBR322. Here you can see BAM H1, SAL1, PVO second, PST1, PVO first, EcoR1, HIN third. These all are the restriction sites, restriction enzymes. So these many restriction enzymes can work on this vector DNA, and it has some selectable marker which is AMPR TTR. AMP is an antibiotic, that is ampicillin, and TT is another antibiotic, that is tetracycline, and R represent the resistance, means those who are transformed cells, those when these vector DNA will make a clone of, it will replicate, then it will make copies of DNA containing a desired DNA with this vector DNA. So how we get to know that our DNA has been transformed or it has been replicated? So to distinguish transformed DNA from non-transformed, selectable marker is used. The selectable marker is ampicillin or tetracycline resistance. If the cells are transformed, so they will grow on the medium containing ampicillin or tetracycline. If they are not transformed, they will not grow, because the transformed cells will acquire resistance. As you can see, they will acquire resistance against ampicillin and tetracycline. So this is used as a screening method. Apart from that, you can see the ROP. ROP codes for the protein which are involved in the replication of the plasmid. So this is the E. coli cloning vector, that is PBR322, which has some restriction sites and antibiotic resistance, and as well as ROP. And this is very important from the exam point of view. So don't forget its structure, and all these things are very important. And we will discuss now. Till now we have discussed about the introduction, how restriction enzyme works, now how the vector DNA looks and why it is important, and after that we will discuss about the rest of the process which is used in biotechnology, so that we can make our desired product. I hope you have liked this video, and if you like this video, please rate this video or rate this course. Thank you for watching.